Hi, everyone. It's really exciting to see everybody here today. Thank you for taking time out of your day. My name is Kira Kappelman, and I'm a first-year PhD student for Dr. Adam Vanyas and Dr. Jean-Ming Yu here in the plant breeding department at Iowa State. And today I'm really excited to introduce our next speaker, Dr. Charlie Messina. Dr. Charlie Messina earned his PhD at the University of Florida before he joined the private sector, where he led research focused on embedding crop growth models within whole genome prediction and received recognition for his extraordinary technical expertise and contributions. Dr. Charlie Messina is currently a professor at the University of Florida in the Department of Horticulture. His teaching and research program focuses on harmonizing crop growth improvement and AI efforts for agricultural systems to regenerate the environment while providing nutrition security, improving human health, and adapting to climate change. He and his team evaluate and apply novel and innovative prediction methods and systems within operational breeding programs. We're very grateful to Dr. Charlie Messina for making the trip to Iowa from sunny Florida. So please join me in welcoming Dr. Charlie Messina.
Thanks very much for that probably undeserved introduction. Certainly has more merit. Thanks for the invitation. It's great to be back in Iowa, even though it's not a bit colder than in Florida. So speaking of cold, this is a hypothesis, really. I'm not going to answer this question directly, but you will see how we can get around and in context of thinking about complex systems as cropping systems, when you tweak some things and say warmer climates, the first thing you think is let's go and do breeding for heat stress tolerance. Well, maybe the system, we can squeeze it somewhere else to get to where we want to go. And perhaps the solution to breeding for climate change is not in the heat side, but in the cold side. So I would like to thank Randy Clark. He was part of my team in Corteva. As you will see, it's been many years on this journey that took us here. And I think it was one of the best things I ever done in Corteva was hire him. So thanks my co-author, Randy Clark. Okay. B73, how many of you know B73? What that number means to you? Hands? So any of you seen it on the a hallway where it says patents and innovations as the greatest things ever done by Iowa State? I think if you didn't see it, I want to put forward a formal complaint. B73, that humble inbred, is, among other things, probably in the background of this drought-tolerant hybrid. And this is a little bit of the journey of the story, where I was taking soil measurements with Dan Bolte many years ago. Those were Aquamax days in Elgin, Nebraska. If you look at that crop, you probably agree with me, is suffering from drought stress, right? That's not very, if you work with corn, that's what corn does. They roll when there's no water. The problem that we face is that we have TDRs in our hands, so we know how much water was in the soil. And if you look at that graph on the right, look at volumetric water content and soil depth, and start from the measurement on 627, 710, you can see there was water uptake, therefore there were roots in there. The 722, the day we were there, you can see there were active roots about 1.60 meters. Why in the world the thing was all rolled up when there was more water than the plant will ever need all throughout the growing season? So that was a puzzling question for us, is we are breeding for drought tolerance and there was drought in that soil. So what are we breeding for? So it was a question that behind me for many years. You see this picture from 2013. At the time, we were also doing all sort of phenotyping to understand root systems in corn. This is a contrast between double crosses and single crosses. We were looking at X-ray technologies. We make these plants grow in systems that with X-rays, we can look at all the details that you can imagine on those root systems. We know there were roots about that depth. So if there are roots, then the question is, are they uptaking water? And we were in California also measuring water content. A number of publications came out of that work with former student Andres Reyes. You can see in this particular hybrid, it's 2.5 meters, okay? They can extract water into massive depths. Corn is competing with sunflower and see who can go deeper. So what is the problem? If the roots can uptake, if maize can uptake water, why in the world are we still seeing this rolling? So that's where we start thinking maybe it has something to do with not latitude, but temperature. So if you look at the Regis paper down about 2.5 meters and 38 degrees latitude, that's California, those are hot soils, warm soils in depth. While some of the experiments, well, the figure you saw, that's Randy Clark's paper, about 42 degrees and not quite a meter and a half. It's kind of Nebraska. You have papers from Iowa State, Ordonez, about the same depth. Some unpublished data was working in Minnesota. The root system were pretty much unfunctional beyond 1.2 meters, and we have 15-minute measurements with some probes. If you look at the literature, Wang, that's in Mongolia. So those are cold soils, right? About 1.2 meters, and you keep going, Canada. So you can build from published data this transect across latitudinal gradient that is pretty much tied to soil temperature. So now the hypothesis that I think is fairly to postulate is that root growth and or function is what, a response to that temperature, is what really underpinning that sort of a drought symptom. So if you want to improve drought tolerance, you better need to look at how these are responding to temperature, not aerial temperature, about 1.5 meters deep. And to answer the question whether we breed for cold stress to deal with climate change, the corollary of not rejecting this hypothesis is if we select for this root growth response to temperature or function and increase the water capture, remember the higher temperature is going to increase that water demand in the atmosphere. So the best thing you can do to prevent rolling and drought is capture more water in depth. So if you can manage to make those root growths in depth and move that water, that's water supply demand ratio become more favorable for the plant, and therefore the heat problem, essentially what you do is you turn out your cooler and your air conditioner by getting water moving from the depth up to the leaves. So that's essentially why the system don't start breeding for heat stress yet. There's some other pathways which is about supplying water to that plant, which is also relevant for today's environment, right? So again, the corollary of testing this hypothesis is what sort of answered that question indirectly. So to do that, one of the things we have to do, and credit goes mostly to Randy, and a little bit of credit for listening to some ideas I had, it was to build a phenotyping platform. But now we have phenotyping built with a very, very clear purpose. You have a clear question that we want to answer. And in this particular case, what we wanted to do is look at root growth response to cold stress. But the story sounds pretty straightforward what I'm going to tell, but we're just bumping on walls to walls, trying different root phenotyping approaches in growth chambers, right? We found that the roots were not growing on the cold, but because the plants were not growing because the cold chamber was at 10 degrees, right, or 5 degrees. So one of the things that we end up doing was we wanted to control the growth of the aerial part so it's not limiting to growth. So when the roots are not growing, it's not because the plant is not growing. So that was the first thing. So you see that sort of a foil covering the tanks, that's insulation. So these roots are going to be cold and the aerial part is going to be warm. The other thing we want to do is the plant cannot grow because the roots are still cold, but we want the growth piece, not the problem with nutrient uptake. So essentially, again, this is system thinking. If the colds are roots, nutrients doesn't get in there, the plant doesn't grow, the root doesn't grow. So you see that sort of black ring along the tank. That's an aeroponic system that is aerial temperature, air temperature. That solution provides a nutrient to the crown of the roots on the top of the plant. So the growth is not limited by nutrients, it's not limited by that, the aerial part, but the water flow is not limited by temperature of the aerial part. So now that system in place and we have happy plants on the aerial part pretty much at the interface. Now we can start cooling the roots tips. So now we can start seeing how they're responding to the cold temperature. So those meristems will be flooded in an intermediate fluctuating temperatures. Therefore, what we're going to see if those roots are not growing is because of a temperature and nothing else. So that was an engineering piece that Randy actually patented to do. The next thing is, okay, now we have a bunch of roots. We can multiplex these tanks. We can run thousands of plants, replications, and whatnot. We need to do some imaging, and that's where the other bit is coming to place. So he built a root system, a phenotyping system, so we can measure for each plant, for each tube, the distance between that crown and the tip of that growing root. And finally, I should have come a little bit earlier, but there it is. We want to know that we answer the question, or we can answer the question to root growth is the function that is compromised here. And to do that, we knew that the root systems can grow very quickly, and we need to get the plants growing up to B7 to get that photosynthesis and have a real plant and not a seedling. So this is a chamber which is about a meter tall, more or less. It's about 4 inches in diameter tubes. And remember, when you pressurize these root systems, right, you want to move the water to answer the question. If you cool the root system, the membranes may be not fluid as they will be when they are at a higher temperature. So you need to squeeze them a little bit more to get that water through. So remember, when you're pumping the bicycle, what thing that happens is your root system is going to get warmer if you get the pressure up. So one of the things is these systems can actually maintain the temperature. So we can cool the root temperature, we can pressurize it, and therefore we can measure the water flow, or at least how much pressure is required to move that water through those membranes at that different levels or different temperatures. And of course, in the aerial part, we don't want to mess with waiting pots and the like, so we have a Lycor system to measure gas and water exchange. There's another, which you will see later, that's for whole plants, but to measure the root or the plant's resistance, because that may still be a problem, we just decapitate essentially the plant and measure subflow. So some of the results you will see relate to subflow. So there's no doubt that the only problem that is in there is the root and the temperature. So what we found, here you have 2 graphs. X is the temperature that these plants were exposed. The y-axis is the primary root growth in millimeters per day there. Sorry about the numbers. On the high end, you probably get about 2 inches, which is what you can get if you grow a plant in aerial outside. That's what you're going to get. So whatever you're measuring outside, forget it. That's not what the roots are doing. What the roots are doing at 1.5 meters or more in Iowa is what is happening at 10 degrees. They're doing nothing. These roots are not, this is a sample of 100 hybrids. Okay, this is not a minor sample. These give us a good sense of what sort of genetic variation or genotypic variation we can find. It's interesting that when we look at the diversity panel, which is the inbreds on the bottom, of course, you would see way more variation in there. You have stiff stocks, non-stiff stocks, material from the northern corn belt and southern corn belt. And the interesting thing is some of these inbreds that can contain some favorable alleles that can push that limit to the 10-degree barriers a little bit low, which essentially is telling us a second fundamental result here is that genetic variance is greater than zero, so we can actually breed and break that invisible barrier that when you dig a hole, you won't see anything. The roots are not there, but why they're not there? Well, there's an invisible barrier defined by temperature, which is that 10-degree temperature. So that was the first finding. So the hypothesis, I think, is holding some water in there. The next piece that we wanted to look at was whether those roots were functional. And here is results to, these are light response curves. The hypothesis from a physiologist is if you cool the roots, the water is not going to flow, so the transpiration and the low temperature should be the maximum transpiration or photosynthesis should be lower at with cold root temperatures than warm root. And that's what you see there. You look at the transpiration and photosynthesis, kind of an awkward units in there is 2.5 milligrams per CO2 at 18 degrees, and you cool those roots to 10 degrees, and you get 1.5. So essentially, what is that temperature of the root system matters, and now we have an assay that we can start discriminating hybrids for that particular trait. So now that we know that the assay is actually functioning and is doing what is intended, now we can start torturing the plant and ask the question, how much do we need to put that pressure in order to maintain the transpiration? So if you, in the x-axis is megapascals, this is how much you need to put that nitrogen through that cylinder to maintain a level of transpiration, say 3. So for a plant which is grown at 18 degrees, is about 2.5 or 3 milligrams of transpiration is about one-ish. Sorry, I would use a pointer, but people in the other side of the Zoom is not going to see a thing. And if you want to maintain that, you need to go to 1.5 megapascals. So when you cool those root systems, and particularly at that depth that I show you for algae, those roots, they are growing little, but they're not functional. And essentially what it does is it requires way more work. Essentially, you need to develop a lower water potential in those leaves. That's what they are rolling to move the same amount of water. The other bit is there genetic variation? We didn't get that far, but we know there's genotypic variation. There's a set of hybrids, drought tolerant, drought susceptible ones in there that they require different, or the subflow is different for a given level of pressure. So, all right, the story is sort of getting together. We see some genotypic variation. So can we map it? And that's, I think, as a geneticist person or physiologist, you're not convinced until you can find some regions, right? Across the 200 lines, do you have that replication that tells you, yep, there's some elements in here and some genes that help you convince yourself that there's some merit in this story? And the answer is yes. We run these materials at different temperatures. We can conduct the GWAS analysis. And you can see that at 18 degrees is the one that Christine Dippenbrook liked to look at that. And her favorite genes was glycosyl transferases or COVID1. She's also excited to call it COVID2. We have some work to do there. But in Arabidopsis thaliana, when you start looking at some of these genes, they're either implicated in ABA signaling or which is elongation deficiencies genes. Essentially, you not only see these genes implicated in the root and elongation of growth, but you can see in the elongation of other organs. A person the other week, I gave a similar presentation, pointed out that these thresholds, they are very much or very similar to what you can see in the leaf elongation. So this may be a control and this response to temperature, which are more broader than just specific to the root systems. What this means. Let's see if I have enough time here. And one thing is, I don't like to get lost in translation, and because it's really easy to get entangled and focus on this pressure and photosynthesis and transpiration, but what really means when you start thinking about developing products. And here is with these process-based models, it's a way to make your list of what make the plant tick and don't forget about anything. And the other bit is, yes, there's this amount of water, but is that amount of water different because we can make that plant move the water faster, or we can make roots grow and colonize that deep layers more? Does it really matters? So this is just a sketch of a model that I use quite a lot to think about within Corteva. I still use it. It is very practical because it has these two equations essentially which are tied together. One is that the variation in growth in time depends on transpiration essentially, and the transpiration depends on the mass production. So depending on how the resources are limiting. Think about this is, as I tell the students, the wolf and the sheep type, or hare and rabbit type of set of differential equations which are tied together. So this system has the potential to oscillate, find equilibriums, or make one thing crash. What it means is that this was, if there's a genotype by environment interaction, is going to expose it. You can think about, okay, let's make the roots grow more, let's capture more water. That's great. The prediction that you may be thinking is we find the solution, it should work everywhere. Hold on. This equation is telling you that the timing and the pattern of water use may be more relevant, and actually that's, or as relevant as the amount of the water capture. And the parameters in these models, some of these are estimated using the GWAS, some are estimated using measurements, as Alejo shows, for harvest index, for example. Some others are estimated based on first principles, depending on CO2 gradients. So now we can use this biophysical machinery. We connect it with the GWAS, so we map some traits. In this case, we use a number of experiments, about 6 years of drought studies. Another called it half-die alleles conducted by Corteva in several locations that we published with Christine. The summary is in that publication. And what we can show is when you combine the crop growth model with this GWAS or genomic selection algorithm, we can have higher predictability. And in another paper, we show we can, when you train these models in a wider range of environments, you can capture back some of the traits that underpin the variation for yield. In this case, you can see the locations. If you add the two first panels in size B, it adds about to 120 locations. These are 140 populations, 7,000 hybrids, 6 years in drought, another 3 years in Midwest testing. It's a humongous data set, and this figure, it tells you we can capture back some of the trait. So we can use some of these machine learning techniques, the old PCA favorite tool. So we can look at these non-Steve Stokes crosses. These are among the diallel. We can focus on these two because this is drought susceptible by drought susceptible and drought susceptible by drought tolerant crosses. So in one case, if you look at that pie plot, you will see one axis that says yield TPE, pointing to the left of the graph, and the another one pretty much vertical, yield water WD, which is water deficit, and very much all this population is high yielding, no drought tolerance. And you cross with the drought tolerant parent, what you can see is that that shifted down, so essentially you can start bringing alleles that contribute to improving the drought tolerance. Now you start looking at the second P-simple component 3, where you can see the root elongation rate more clearly, kind of on the vertical axis in the middle, RER, and you can see that this yield resistant or non-drought tolerant material, they have average or low root elongation rate. And when you do the same thing for the drought tolerant parent, you can see how that high population just shifted on PC3 up, and is all pointing to the root elongation rate. So again, now we have hypotheses that now we can identify germplasm, that we can go back and test it for whether it's the occupancy or the function of that root response to temperature that is really underpinning this. Essentially, we use that function in the crop growth model, trying to rescue back what may be explaining the genetic variation that we have in the germplasm. Because we train the crop growth models for some of the traits, what we can do now is we can exercise the model and ask the question, is if we increase that growth under low temperature, let's say by a degree, or the slope of that by a degree, or increase the conductance, what is the likely impact that you can have? And you can, we can do the two at the same time. We did this for a number of years from 2012. You remember the drought in Iowa, that was Aquamax days. 2016, there was less of a drought. And you can see that the frequency of that blue, it changes depending on the year, obviously because the water limitation is different, obviously different in space and different between the different traits or the trait combinations. So let me focus on something that I pick is in the 2012, that I just want to bring your attention that we can increase the water flow, we can increase the exploration of root, but you can still have negative impact. You can see that sort of a part of western Minnesota, northern Wisconsin is red. We increase the water capture and the yields go down. Why? And this is not necessarily something intuitive. And the reason, after thinking and bumping in walls long enough with empirical, which is what we learned from Aquamax, is the timing of the water use. You increase the amount of roots and you put the roots in depth very quickly, you let the water flow, and the plant is going to grow during the vegetative period. The harvesting is going to be very low, and it's not going to yield. So essentially, some of that traits are kind of iffy to work with. You need to get the right balance. But by large, for the rest of the Corn Belt, what essentially is telling us, if you can increase that root growth, is going to have a massive impact everywhere. And essentially, if you think about that sort of isotherm of 10 degrees, if you measure that in Iowa, you're going to see about 1.5 meters. Just get another 10 centimeters, and that is a massive mass of water, and that's yield stability. So to deal with the climate change and the heat, probably need to start breeding for the cold. The other thing we can do now, thinking a bit harder about climate change, is constructing some profiles, and you can do this for any genotype that you have information, where you can, instead of using Philly and Wilkinson and your environmental gradient is on the x-axis, we can put an yield, and then look at what traits explain the variation in yield on the simulated values. Each of these colors correspond to a trait. I just want to make you aware of three of them. One is that sort of a brownish, which is the size of the ear leaf. There's many posters in here. Size of the ear leaf is a great thing when you have water. So explain a lot of variation in 1500 gram per square meter on the 270 bushels or so. The other one is the root-based temperature, that sort of purple, and the dark green, which is the root traits we are interested in. So now that we have an idea that the roots matter between that 500 to 1200 and size of the ear leaf is in the higher end, we can start looking at where the Corn Belt sits. And of course, consistent with the maps, those roots essentially explain a lot of variation where it really matters for corn breeding. Climate change is going to push this box to the left, simply just the merit of increasing temperatures and water demands. So if the shift in some locations go from environment 2 to environment 1, because the trait is about increasing that yield, think about, we have a smaller plants, but it's not going to generate genotype by environment interaction. Smaller plants are going to yield less than bigger plants. That's what some of the posters are after in here. If the climate change push environment 2 to environment 1, and now the different traits are really mattering for the adaptation, now you can start seeing lack of correlation and crossover interaction. So breeding for climate change is not just, again, the cool roots or the heat stress. Each environment is going to find its own combination of traits that is going to really matter. And this is a type of technology that you need to get to start thinking about how to take that long-term genetic game machinery and start steering to the right directions. Yes, and the other piece is never forget that the correlation that is most important is not the accuracy of your correlation of your prediction method, it's the correlation between your multi-environment trial and the target population of environments. You can have the perfect predictive machinery, but if you're predicting the wrong TPE, you're not going to make a lot of genetic progress. Just a message for the students. So now we can start routing a solution. I think root and function underpin that water deficit symptoms. I convinced myself, I hope I convince you. There is significant genetic variation for that root elongation rate and the low temperatures, so there are scope for breeding programs to harness that. There's genotypic variation for root conductance. There's a lot of work to do there, but it's promising in my mind. Both contribute to yield improvement. And I think as we think about these traits, I think it's going to have great importance on the warmer climates to rebalance that water use. And it's not just push the whole thing to an extreme. It will have to be balancing that water, the water supply demand over the growing season in order to get the right patterns. And that's one of the key traits that underpin the success of Aquamax or the previous success of Aquamax. I lost track of it. So thank you very much. I will acknowledge all the support from Corteva to many years. Christine, Leo Perugini, it was a pleasure to work with them doing genetics, Tom with statistics, and of course my partner in crime, Mark Cooper. And we are looking for postdocs, so send me a note. I can't hear you.
In terms of the application in an agricultural drought or a meteorological drought compared to the types of tests which I think are more on the hydrological side of things.
Yeah, this generates a little bit of the confusion. Think about the drought I started, that's the first picture, that's agricultural drought.
Yep.
What we did in the experiments were pushing the plants to expose genetic variations. Essentially, we're trying to dissect the thing. And then we put it back together within the crop growth model so we can assess the impact of that trait from non-agricultural drought, let's say in Colorado, corn is not going to grow well there, to well-watered environments of Ohio. So you see the maps across all the range. So we can then assess the contribution of these traits in the environment that matter to you. Actually, I like your statement, non-agricultural drought, yes, on the 100 millimeters of water, you're not going to grow corn, right? But you saw that in the, 1,200 grams per square meter, that's a probably 300 to 400 millimeter environment. So that's not agricultural drought. And you can see the root elongation really matters in there.
That's good. In a follow-up, if this was not a maize, like as a crop with the warm season, and what would you propose or hypothesize if it was a different, was cool season grass or, some of the crop that probably is, originating in a different kind of climate?
I can tell you, we are looking at introgressions from highland maize because we think there's some segments in there that can increase the root growth under low temperatures. I think looking at different species, you can learn for gene editing opportunities. If it's corn, I would start screening for this thing because in the northern environment, that sort of bucket is much smaller, right? If you look at some papers in Canada, I think it's 2 feet. So the drought can be way more frequent and herbaceous, even though any day the soil can be wet, but can be dry 3 or 4 weeks after. Same thing as Florida, right?
I worked in Canada, so
Yeah.
I understand what you're saying. Thank you.
No, thank you.
Thank you, Charlie. It was really nice talk, thought-provoking. So my question was about how much variation in temperature along the rooting depth do you see with the soil type? So in other words, is water holding capacity, I mean, water's got a lot of capacity to store heat and lose heat when the temperatures are different between air and soil. So how much variation,
You're
you
going
have two
into a very
locations, one
detailed wrinkle here that I don't remember how much is the gradient.
but does
But-
soil type, do you think soil type and temperature would matter, or is it?
Oh, so yeah, soil type, of course, yes, is going to affect the soil water, the thermal conductivity and the soil water holding capacity and how the roots, the depth are going to hold.
In
right?
terms of temperature?
Sure, yeah, absolutely.
So the
Tirius is there, he can do the calculation for you.
other question I had was, for kind of going more towards the technical side, when you had the setup, how much of the conductivity of water, water conductivity in the roots is inhibited by, cavitation and other things that would presumably get driven out when you apply external pressure?
How much is cavitation? No, I don't think it's cavitation because the water is limited by the membrane conductivity, not by the
taking
pool.
out. Yeah.
Essentially, this is how much we have to pressurize the root system to get that water in the soil column through the roots to the collar. So there's no, I should have stated that these are water columns, right?
Yeah.
The roots are there, but they're just cold.
Thank
So
you.
again, at 10 degrees, so they're not growing and you need to pressurize up to 1.5 megapascal just to make that water cross the membranes.
Yeah, thanks.
Hello, thank you for your presentation. I was thinking about the plot you showed about the hybrids, the virtually and invisible barrier at 10 degree. That plot was done at different exactly temperature control. Do you have a treatment that is below 10 degree, or was that also the same as a 10 degree, 18 degree, and 25 degree?
This one,
Yes.
yeah, these were below 10 degrees. I can't remember
You had
exactly. I
a
think it
data
was
point,
5,
10
10, 15.
Then you ran a curve, smooth out.
Yeah,
the
exactly.
treatment.
Yes.
And that's great to know. And then also, you mentioned about system thinking, and you build this aeroponic system, supply the nutrients and hydroponic roots. And how do you think that's simulation setting is corresponding to natural field conditions?
Right now,
Because there, at one part, you want to dissect the roots and shoots that to remove that interference. But we also know that root is sending the signal up, shoot is sending the signal down. How would you think that, like further improve that system to get more findings? Yeah.
Yeah, I think it is similar to the answer before, right? We use this system to dissect and understand the trait. Now that I understand the trait, the same thing we did in, let's say, drought, right? You figure out that silk elongation really matters. You're not going to build a silk elongation platform to select for. What I think we need to do is figure out a way to put this germplasm in a gradient of soil temperatures and let the drone tell you what are hot and what is cold, because that will tell you the water is moving, it's capturing the water, the roots are active. So I think it's a difference between the phenotyping to understand the physiological genetics in the absence of confounding relative to what is the screen that we are going to implement in the field, which is high throughput and we can fly a drone or go with a combine for that matter. So I think in the two different things. One is understanding, the other one is selection, kind of a managed environment for selection, right? And we can calculate the correlation between the two, which I didn't do. you can look at the genetic correlation. Maybe the control environment system is more appropriate to do.
Great,
My
thank you.
experience is working in woodland. You're better off having your field trial, do the right management, and that's your highest throughput phenotyping possible. As long as you know what I'm talking about. The correlation between your multi-environment trial and the TPE is high. We are good.
I have another technical comments or questions because I've got to see this slide twice. When you show the GWAS hits under the 10 degree, 18 degree, and 25 degree, I can show that slides quickly. I did a quick visual summarization. It looks like there's more overlapping hits between the first two temperature, and there's more for the last 18 and 25 degree. So it seems like certain genes will show its effect. Temperature reach certain stage. Otherwise, you won't detect any signals. Was that a good summary? Or?
That sounds about correct.
Thank you. Thank you.
Charlie, thanks for a very great presentation. I have lots of questions, but one clear message from your talk, it is all about water. This is how I take a message from your talk. And my question is about perspective for the future. Do you think changes in the root system or a little bit deeper roots will be enough for us to get into the future, or we will need more drastic changes or incorporation of irrigation or something to maintain this yield potential that today's hybrids have?
It's a long shot to speculate about. What I can tell you is that I think the future is more than water. The story starts with water, but remember, nitrogen moves with water, right? So if you cannot move water through that nitrogen in Nebraska, it was staying there or ready to be moved to the groundwater, to the water table if there was no uptake, right? So that's one. The other thing that puzzles me sometimes in Iowa is how much nitrogen moves below that 1.5 meter or whatever feet that the corn cannot recapture because the roots are not there. So that's another thing. I think water and nitrogen, those two things go pretty much hand in hand in my mind. So this is a good trait at least to shoot for. Climate change is going to rearrange the map of crops. It's been known for many years. So I think we need to be prepared to say, okay, we are going to stop growing this corn in these areas because whether it's too hot or whatever, we're going to be replaced by sorghum, or maybe these new areas is going to open up, like in Canada, for example. So I don't know how it's going to look like pattern, but definitely it's going to be a rearrangement of regions, and breeding programs are going to adjust to that. The question is how quick. That would be I don't know, irrigation, I don't think we have enough water, frankly. I wouldn't count on that.
Thanks.
That's all the time we have for questions. So let's think. Oh, there's one more. There's time for one more.
The boss is there, eh?
So this is a question that we have online. Can you hear me? Oh. First of all, thank you for coming to Iowa to visit us and for giving this nice overview of cropping systems and system thinking as you and Yamin were discussing, because that is something that is super valuable for us, and it's also super valuable for us to have you, so thank you for that. But I have a question online. So the question is, I wonder if you could explain more on how you have obtained the yield variation, explaining for each trait. The figure, one of the latest figures.
Imagine this figure.
I think so, yes.
Yeah, this is a simulation study. It's a large-scale simulation study that you have control over all the traits. You can do the same thing with empirical data, but it will take, I don't know, maybe some of the era hybrids that they were growing and they have multiple traits. You can probably construct something similar with empirical data. for this study, we would really want to build some theory and thinking. It was just a simulation.
Also, for that one, you mentioned something like, let me see in the notes, that the environment kind of choose the right traits, right? But there are certain environments prefer certain traits that can explain better either, right? My question is trying to twist this a little bit is if we know that certain environments prefer certain traits in other words for improving yield, what if we redesign crops thinking on those specific traits for those specific environments?
Think about that sort of box is your target population of environments, right? And if you're thinking on Nebraska maize hybrids, move that box to the right, and it's a lot about growing big plants and fast with high radiation efficiency, a lot of nitrogen in the leaves. That's pretty much what you see there. Move that to the left, and you have that ear size at silking. That's about silking elongation, reproductive resilience, get those silks out of the husk as effective as possible. That's Aquamax. Of course, the roots we don't know quite a lot, but some of the radiation use efficiency is not the highest because they're conserving water. They have the limited transpiration trait tied to the radiation use efficiency. And they're not the biggest plants in the lot. You can use plant population to tune it to the different water availability environment. So maybe another message here, and this is yield environment and traits. This should be three-dimensional. I need to have a agronomic management, plant population to one of the sites, but I think you can think about what is the mixture of environments in the target population of environments, and then you can start thinking about what traits can give you a combination to succeed and get successful hybrids in that region. Not an idiot.
Thank you.
I think that's all the questions for Dr. Messina. So please let's thank him again for his wonderful presentation.